Vale, pues Tony, cuando estés listo,
Sí.
dejamos unos minutitos que se está uniendo la gente, pero ya podemos decir un solar. ¿Estás escuchando doble también, Tony? Vale, se ha arreglado mi error de la doble voz. ¿Puedes escuchar bien, Tony, ahora?
Tengo.
Vale, es que te he escuchado doble. Pues a ver, que compartimos el link. Vale, pues Tony, cuando quieras, todo tuyo. Estás en mute.
I will share the screen and start the presentation. So, good afternoon, everybody. I am Seminarios Martinez, PhD at the Ancient DNA Lab in Tartu, Estonia, in the Estonian Biocenter. And today I will present one of the projects I did during my PhD thesis, which was the retrieval of Salmonella enterica paratifici sequences from one soldier of the Barcelona siege in the 17th century. This is the following structure I will follow in this presentation. I will present the introduction, a brief introduction about the field of ancient DNA, a brief, also little brief methodology that we use in ancient DNA studies, which are a little bit different than other genomic studies, the main objectives of this particular project, the results, and some brief conclusions that we can extract from it. So the first thing that we have to get in mind is that from the perspective of ancient DNA, humans and pathogens, it's not that right now, I mean, it's a really hot topic with COVID and monkeypox, but this relationship between pathogens and humans has existed from millennia. I mean, in fact, since the emergence of humans as a species. And we can find signs of the adaptation to those pathogens. They have represent a really strong selective pressure to humans. So we can find the signs of molecular adaptation in humans to those pathogens. And most of the pathogens that we know today that causes disease, for example, flu or hepatitis B, they're associated to the Neolithic transition, which basically it's a social change in human populations, in which if we start from hunter-gatherers populations before the discovery of agriculture, hunter-gatherers were living in a really sparse, really sparse populations in really big territories, and they were not staying at the same place most of the time. They were just looking for animals to hunt and get meat. Basically, during Neolithic transition, what we get is three factors. The first one is that humans have to stay in the same place all together, and at the same time they're living with animals that these animals were before roaming in the nature, but right now people are raising those animals for meat. So the animals are living really closely together to the human populations, with a constant source of resources. Human populations grew a lot, so we have really dense populated we have really dense populated populations, and also we have to start to think that those people, they were not taking care of all the residues that they were generating. So they were living with animals, with a lot of with excrements, with stale water that was really close to the place they were living. Those conditions propitiate that some of bacteria that were pathogenic for animals, or some of the bacteria that they usually inhabit in the feces or the residues of a person, now they have the chance to jump to people and extend across the population. From an evolutive point of view, pathogens are really strong selective pressure. We can imagine that certain populations have different alleles for a gene. We would say the green allele and the blue allele. So the pathogen, if a pathogen infects somebody with a blue allele, we can get to the point that that person cannot reproduce or that dies. So that gene will not be passed to its descendants. While if they are carrying the green allele, these people are more likely to survive and then just pass this variant to their offspring. So we can see that there's a clear selective pressure, and certain genetics patterns can emerge from the interaction of humans and pathogens. As we see with time, if we start from a population with there are balanced frequencies of these alleles, we see that with time and the selective pressure exerted by the pathogens, we purge most of the population that carries the deleterious alleles, and at the end, the population is carry only the allele with good fitness. But this is not only true for the human populations, but also the pathogens that are infecting us are subjected to those selective pressures. We have to have in mind, for example, the immune system. Our own immune system exerts a really strong selective pressure to those pathogens. So, and also other factors that in this case is, I think, quite hot topic. Antibiotics, they also suppose a really strong selective pressure for the pathogens infecting us. So with time, for example, in the case of antibiotics, what we have is that the contact and the constant exposure to those antibiotics select the pathogen populations to be resistant to them. So why why do we use ancient DNA to study pathogens? The first one and more clear is that since most of the pathogens do not leave a clear physical evidence in human remains, archaeologically speaking, testing for the presence of the DNA on those pathogens can help us to shed light in past epidemics, or, for example, epidemics that we don't know the origin. With this tool, we can learn which was the most likely pathogen. But they are also really important because they can be used to calibrate phylogenies, in the sense that if we assume that the time sampling, so when the bacteria or pathogen was living, it's directly correlated with the number of mutations that is accumulating in its genome, we can predict or we can date when different populations of pathogen splitted. And can be also really important just to try to look of genetic affinities between pathogen populations in different parts of the globe. And finally, related to selection of those pathogens, we can use ancient data to see how a naive pathogen, a pathogen who has not been exposed or to humans, so a pathogen that just emerged from animals, or how a pathogen was before the introduction of antibiotics. In that case, this has been successfully used. In this case, this is the case of Native American populations during the discovery era. Basically, there are a series of outbreaks of epidemics that drastically reduced the populations in the Americas of Native Americans during the Spanish conquest. And we know, for example, by chronicles, that some of the diseases were known. Smallpox is one of those examples. But in this case of the Coevolution, those until really recent, those outbreaks were of unknown origin. Now we know that those outbreaks were associated to Salmonella enterica. So this ancient DNA is a really powerful tool to get more resolution in order to discover ancient epidemics and their origin. Also, one of the most paradigmatic cases is the case of plague of Yersinia pestis. Basically, thanks to ancient DNA, we know almost step by step how Yersinia pestis emerged from its natural reservoir in rodents, and it started acquiring more virulent traits with time, which propitiated in the peak historical pandemics during the Justinian plague, and the Black Death, and the Third Pandemic. But also, it helped to discover previously unknown epidemics during the Bronze Age and the Neolithic. So it's a really good tool to discover things that could not have been written in history books. So I think it's quite characteristic of ancient DNA to study ancient DNA that we have to follow a certain process. It's not they are not as a standard as you would use for normal genomic studies, let's say, with modern DNA. Basically, this is laboratory-wise, this is a typical scheme that you do. You extract samples from one human source. Usually, depending on the type of DNA that you want, if it's for the host or for the pathogen, you extract the DNA from different types of bones. But usually for pathogens, we use tooth root. Basically, if a pathogen becomes septicemic, it is released in the bloodstream, and it can be preserved in the root of the teeth. But you can also use different non-so-conventional samples, for example, medical collections present in a museum, that they know that they are originating from an epidemic, a historical epidemic. Those can be also used as a source of ancient pathogen ancient DNA. So DNA is extracted and basically sequenced, and well, it has to meet certain quality thresholds. But if those thresholds are passed, we go to the more bioinformatic part. And to understand ancient DNA, and how to work with ancient DNA, we also have to take in mind its particularities. And there are basically four. The most characteristic ones is that there is a really low endogenous content of DNA, both for host and for pathogen. So this is an example of, this is a real sample. Basically, if you sequence, I remember there were here around 200 million reads. Most of those reads, they are not represented in metagenomic databases. So we don't know basically what they are. But again, those reads were originating from a Petrus bone. So Petrus bone is a really good source of human DNA. Again, most of the DNA is not corresponding to the host. It's just soil bacteria or fungi. And only a tiny sequence, it's 3%, it can be directly associated with the endogenous DNA. Really related with this is contamination. We can be sure that the DNA maybe is human, but the problem is that maybe these bones have been handled by archaeologists and not in the best conditions. Since there is so low amount of endogenous DNA, any kind of modern contamination will be also reflected on the sequences that you're retrieving. So you can say, okay, I have an Neanderthal bone and I could recover 3% of DNA, of Neanderthal DNA. But if you take a closer look to this DNA, you will see that in reality, only 1% is truly from the Neanderthal individual, and 2% is DNA of all the people that handled this bone during all the extraction and process. Also really associated with ancient DNA is damage patterns. So basically, because of the chemical properties of DNA and all other external factors such as humidity, temperature, and chemical background and chemical composition of the soil, DNA starts breaking. And also some of the bases, we call it deamination patterns. Basically, some of the bases lose the chemical properties. And for example, if in your read you have acetosine, and originally you have acetosine, with time this read breaks down and the cytosine becomes an uracil that will be read as a thymine during variant callings. So this is good for a point of view, because you can use those damage patterns to differentiate truly ancient DNA from contamination in your sample. But at the same time, if you're trying to do variant calling and analysis with the genotypes of the things that you have sequenced, it can hinder those analyses because you're getting SNPs that they are not really there. They are just damages at the end of the reads. This is an example of one of those damage patterns. So basically, you have to imagine that you have a DNA read here, and the more you go to the end of the read, because of chemical properties of the DNA molecule, cytosines are degrading into, in the 5' end, it's cytosines to thymines. Basically, here we have, for every cytosine that we have in our reference genome, 25% has become thymine. And in the other case, if we are looking at the 3' end, we will see the same pattern, but instead of cytosine, it is guanines that have become adenosines. And related to this degradation of DNA, it's the complex topics of preservation and studies. I mean, here we enter also in the kind of, it's also associated with social differences, not differences, but the word is disequalities. So basically, one, rich countries, they can afford to study ancient DNA. So for sure, as we see in the map, those are, in 2017, all the human samples that have been retrieved from archaeological samples. Basically, rich countries can afford to do ancient DNA studies, but also cold latitudes are better for DNA preservation. So you also will expect that northern latitudes with colder climates would be better, more suitable for the conservation of ancient DNA and then for retrieving DNA for studies. This is those things that examples that I have explained. They're common to all ancient DNA studies, but in particular, for pathogens, we have also some kind of other difficulties. It's that the first one is that most of the time, pathogens do not alter the physical structure of a human remain. Basically, you don't know what has ancient pathogen DNA inside. And this can be solved by doing massive screening. And we are now talking about studies that they are screening in the order of hundreds of thousands of samples just to retrieve several pathogen genomes. Also, again, the metagenomic composition of those samples, it's really variable. And the amount of pathogen real ancient pathogen DNA, it's really small. So we have also to consider that most of the time we will have to retrieve very low amount of pathogen DNA, and even if necessary, we will have to capture the genome to try to retrieve it in its completion. Another difficulty to the variable metagenomic composition of ancient DNA samples is that bacteria there can look really similar to each other. So, for example, in the case of Yersinia pestis, you also have another. Basically, they are a clonal species. It's Yersinia pseudotuberculosis, but it's not as virulent. It's also a zoonotic disease, but it's not as virulent. If you are not careful, you can mistakenly assign rates of Yersinia pseudotuberculosis to Yersinia pestis and get false positives. So it's also we also have to be really careful when we try to ascertain the presence of a pathogen in a sample. Another other problem is that what we usually use is to get to map our sequences against a reference genome. The problem is that we are only going to recover what we know that exists. For example, if a pathogen contains a virulent gene that is not present in modern populations, we're not going to recover it because we don't have reference to map it. But finally, I think this is more a really good advantage. And as I said before, pathogens are really useful to calibrate phylogenies. So if we don't get this proportional distance, mutation distance in a tree, using only modern samples, if we add our pathogen at the base of the tree, usually, and it can depend on the organism that we are using, but usually what we can achieve is to get a temporal signal. So a correlation between number of mutations and time of sampling, which can help to date the split between different branches in the tree. So the main objectives of, now that we have seen all the particularities of ancient DNA and the things that we have to take into consideration when we are working with it. The main objectives of this particular project were basically from a human genetics point of view, try to know which population served in the Spanish army during the 17th century, just more like as a curiosity. But then try to find evidences of a plague epidemic that they were recorded in chronicles, in contemporary chronicles. So it was by historians, by chronic, but people living in that era, it was reported that there was a plague outbreak during 1652 in Barcelona. So we wanted to try to retrieve Yersinia pestis sequences from the samples. And finally, we found Salmonella enterica. I mean, it's a little bit of a spoiler, but we find Salmonella enterica sequences in those particular samples. We wanted to relate those particular sequences to other historical samples, in the case, the ones of the coccolizli, of the Mexican epidemic during the Discovery Age, but also to other historical and contemporary strains of the bacteria. So as a result of this project, I'm presenting the result of Salmonella enterica from a soldier from the 1652 siege of Barcelona, support historical transatlantic epidemic context. Basically, during the construction of La Sagrera station, there were found several burials, several pits with a lot of human remains inside. Those pits, using archaeological context, for example, coins or the type of uniform that the troops were using, has been dated back to the Reapers' War in the context of the 13 Years' War. It was the fight. Basically, the Principality of Catalonia rebelled against the Castilian crown, and there was a big fight. They sieged Barcelona for 2 years, and at the end, the Catalan troops they surrendered themselves. It wasn't particularly assigned to this war because in all the pit there were hundreds of human remains, but all of them they were males in age of fighting capabilities, so between 16 and 40 years old. And we presume that this type of burial can be associated to an epidemic because there was no signal of violent death. No, there were no bullet holes, no fractures. They just were a lot of bodies buried in this pit with a uniform. Usually in war, you just get the uniform. Everything that you can take profit of it, you will take it from you. It's not normal to bury people with their uniform and the weapons. So from those burials, we selected a set of possible individuals, and we could retrieve DNA from 2 different individuals using shotgun sequencing. And we performed human population genetic analysis of those samples, but we also screened the metagenomic context of them. From the human genetics, this is a PCA. So basically, this is representing a current Western Eurasian genetic diversity in humans. And if we project the genetic data of our samples, we see that they are clustering together with Basque current Basque populations and current Sandinian populations. These more or less indicate that the most probable origin of the army was Castilian and not Catalan, because at that time the Basque Country and the island of Sardinia were part of the Castilian crown. So more or less we can say which was the origin of the army. I mean, we did not proceed with further analysis to confirm that. It is just quite exploratory. We also took a look at the mitochondrial haplogroups, and they are also corresponding with Basque populations and Sardinian populations. For the metagenomic screening, what we see is that there are not much sequences that could be directly related to Yersinia. Those numbers seem quite big, but if you blast at the end for metagenomic studies, blast is the gold standard analysis. We see that most of those sequences cannot be assigned directly to Yersinia pestis. In contrast, we find a significant amount of Salmonella enterica sequences. Now that we know that the possible pathogen is Salmonella enterica, what we did is first try to put our sample in the context, in the phylogeny of all modern Salmonella enterica strains. And also we tried to decide which was the closest strain by mapping against all possible reference genomes. And we decided which was the most suitable reference genome by a set of different statistics, namely the distance and the number of mapped reads. So we mapped against these 800 different reference genomes, and we see that the most suitable, in principle, the most suitable reference genome, the serovar, the most possible serovar is ParTPC. It has the most amount of reads mapped against this genome, and also the reads are quite similar to the reference. This is a representation of the ParTPC reference genome, and in this inner ring, we see how our reads are mapping against the reference. So we see that the reference, the distribution of those mapped reads is quite equal along the reference genome. If it was not Salmonella enterica, we will see that, for example, only the reads are only mapping in this region, and all the rest of the genome is basically empty. And we check also the deamination patterns to prove that there is not a laboratory artifact, that somebody was sequencing also Salmonella in the same laboratory. So we see that those reads are truly ancient because they are presenting deamination patterns at the end of the reads. So now that we know that ParTPC is the most plausible agent, what we did is to try to place it in this phylogeny called the ancient European superbranch of Salmonella enterica, which is basically a conglomerate of different zoonotic Salmonella enterica serovars. We try to locate it because those mapping analyses, they are not completely precise, and our sample can be falling outside this tree or in a different serovar. So we have to be sure that it's Salmonella enterica ParTPC. So this is the whole diversity of serovars in this particular superbranch. And here we have Salmonella enterica ParTPC. And in our case, this is our Sagrera sample, and it's falling just basal to Salmonella, to the modern ParTPC clade. This is a good indicative that it's ancient, that it has not accumulated mutation, so it's falling within the tree, but at the bottom of the tree. If we get a closer look to this phylogeny, we see that all the modern strains of ParTPC are in the most outer branches, while our sample is failing basal to other historical ParTPC strains. In that case, they are the ParTPC that causes the Mexican outbreak in the 1500s. And more basal to that, there are other medieval ParTPC strains. So now that we have a really high support of our sample in the ParTPC phylogeny, what we did is to try to remove all possible errors, that errors mainly homoplasia and recombination events, which can alter the shape and distances of the branches in the tree. And we tried to do a typically rated phylogeny in order to know when our Sagrera sample split it from the Mexican samples. Here is what I was commenting before. This is we find a correlation between the sampling date, so the age of the strain, and the root to tip distance, so the genetic distance between the base of the tree and the top of the tree. And we see that there is a good correlation in that case that can be used to explore the dating. So we perform the dating analysis, and we see that around the 14th and 15th century, the Mexican and the Spanish Salmonellas split in different branches. So this gives us a support that is that this, I mean, it's quite how to say, it's quite probable that Salmonella arrived to Americas from Spain. And in particular, we found a really good candidate for an original strain for Salmonella in the Americas. This recent split between the Mexican and, as I said, this recent split of the Mexican and the Spanish strains can also mean that Spain is the original source of those specific strains, but it can also mean that people who went to America and got and were infected with Salmonella could also went back to Spain and spread again the disease in the continent. So, as a brief conclusion of this study, the first one is that regarding the human DNA and the soldiers, we can say that the origin is compatible with the Castilian army because of the region, the Basque Country in Sardinia. The second conclusion, quite big conclusion, is that although we cannot retrieve Yersinia pestis sequences from the site, we were able to recover a partial genome of Salmonella enterica. And the final conclusion is that the tip-calibrated phylogeny is compatible with a recent exchange of Salmonella strains between the Americas, namely the epidemic strains of the Coevolution and the Spanish strains during the Reapers' War. So thank you very much for your attention. This project has been under the supervision of Carlos Zulueta Fox in the IBE. And right now I'm supported by a grant of the Estonian Biocenter and an ERC of the Ancestors Project in collaboration with Cambridge and La Sapienza. Thank you very much for your attention, and if you have any question, I will be glad to answer it.
Thank you very much, Tony. Yeah, some people, I don't know if you can see it, but some people is clapping to you with some emojis. I like a lot of emojis of clapping. So I think you did really well. This is what they want to say. And also I see one hand raised, like from Pablo Carrion. Yeah, I can see your photo. Can you unmute yourself, or do I have? Yeah, you can. Perfect. So
can you hear me already?
Yes, I can. Tony, can you?
Yeah.
Okay. Yeah, Tony, thank you for the talk. It was really, really nice. And I only want you to raise a question which is a little bit divergent to the process that we used, which is what is your, because there are much more methodologies in retrieving ancient genomes from pathogens. What is your opinion, or if you can give a little insight on capturing techniques of ancient pathogens?
So basically, in that case, the signal was so clear that we were not using a really accurate software. Kraken 2 is not specifically useful to get positives. Nowadays, there exists a more precise software, such for example, Malto Kraken Unique. So I think that the insight will be to just use something really precise to discover the lowest amount of reads possible. So, for example, I know people that just with between 50 and 10 reads, they can ascertain the presence of a pathogen in a sample using the software. And afterwards, the idea will be shotgun sequencing is not really efficient. You have to sequence a lot to just retrieve a little bit of coverage of the pathogen. So the idea will be to try to find a possible candidate, pathogen candidate, and then create maybe a custom capture array to try to retrieve as many possible sequences as you can. And afterwards perform, capture-based methods they're really efficient. You can just retrieve in the order of tens of folds of coverage with them. So I think it is the best approach to use.
Okay, thanks, Tony.
I don't know if there is more questions from here, the audience. We also have people in YouTube on live, but right now I cannot see any questions. I have some general questions, Tony, maybe if you would like to answer. I forgot to mention that this is the work you did during your PhD thesis, It's already done. So you spend a lot of time and effort to do this. So first of all, I would like to know if maybe we have someone in the audience that is going to start the PhD or is thinking how to do the PhD, how you reached this laboratory of Carlos Lalueza, and what was the topic that passioned you to lead or to start this PhD with them?
I mean, I think it can seem quite silly at the start, but basically I liked a lot a lecture that he gave us about Neanderthals. And then it was during the master's degree. And at that particular moment, I didn't have any laboratory to do my master thesis. So I asked him if I could join his lab to do the master thesis, because I was really interested in Neanderthals. And he said, oh yeah, okay. Right now he didn't have any Neanderthal or human sequences or studies, but he was starting to do these pathogen studies. And I stayed there for the master. I liked the laboratory. He offered me to do the PhD with him, and I accepted. I think it was a really good decision.
That's good. And is Carlos Lalueza like a bioinformatician lab? Because I'm seeing like a lot of laboratories that are clinicians at the first, so they are expanding to the bioinformatics, and because this is a bioinformatics student group, I would say, how was your experience with Carlos doing the bioinformatics? Like,
no, Carlos has the wet lab background, so he was working in laboratories. But I was really lucky that although, for example, he was not coding, for example, he understand the concepts. Like, he was really wise in that sense. He understand what I did and the purpose of it. He just didn't know how to do it himself.
That's good. And my last general question, which was the biggest problem maybe or impediment, like due to the topic of your PhD? I guess you were sometimes having some problems regarding the samples collection or the number of samples that you got. So how you deal with that problems?
Yeah, the thing is that stochastically, it was quite difficult to retrieve pathogen DNA from the samples. I had the luck that there were a lot of samples going to the lab all the time, different things, and I could choose what to screen and what to get. And I think as another problem, for example, in this particular study, we wanted to perform capture and sequence more, but we got the problem that it was in the middle of the COVID pandemic. Like, we started just before the COVID, and as soon as we get the more preliminary results, the pandemic started. So basically, our archaeologist that was collaborating with us got really sick. We couldn't get in contact with him. We couldn't get more samples. So it was quite chaotic. And at the end, I had to finish the PhD. And at the end, we decided to just use what we had at the moment and pursue with it.
Yeah, and that's another question, like just curiosity. Have you ever been involved in the collection of these samples or any of the samples of your PhD or other samples?
Not really. Yeah, I am one of those strange persons that do ancient DNA but just stays in front of the computer. And no, not really. The closest thing that I've done is go to a museum and see how they extract the things in the museum, but that's it.
Well, it's an experience that you can do later on, now in your postdoc. But yeah, I think it can be great to take some samples.
I would like to go to a lab at some point.
Sounds good. Then, well, I don't know, I cannot see any more questions. Let me just check the chat. Perfect. Then for the YouTube audience, I'm sorry if I'm not able to see the questions, but you can contact, I think Tony put the email in this slide. So if you have any more questions, you can contact him. And thank you very much, Tony, for your time, for presenting your PhD thesis, and congratulations for your work.
Thank you.
And well, for all of the people here, see you on the next Bioinfo Field, which is going to be the next year, so in January. So Happy New Year to everyone already. Bye.
Bye.